Hi, my name is Linda Ma, and I did my chemistry project on protein structure and enzymes. So some background about proteins. They are long chains of amino acids, which are difunctional molecules containing a basic amino group and acidic carboxyl group, as you can see in the figure in the upper left. And amino acids are linked by amide bonds to form these proteins. And then protein classification is based on the 3D shape, which affects the protein's characteristics and structure. First category of proteins is fibrous proteins, which are polypeptide chains arranged side by side in long filaments. And because these proteins are pretty tough and insoluble in water, they are used in nature for structural materials. So examples of fibrous proteins are things like collagen and connective tissue. There's also globular proteins, which are coiled into compact spherical shapes. Unlike fibrous proteins, these are generally water-soluble and are more mobile within cells. An example would be enzymes, which we will go over later. And yeah, the figure in the bottom left just shows just a random protein, and you can see all the different components of it and its 3D shape. Since proteins are very large, describing protein structure is best accomplished using 4 levels, which would be primary, secondary, tertiary, and quaternary. So for primary structure, that is just the amino acid sequence of the protein. And then for secondary structure, that is the orientation of segments of the peptide backbone in a pattern. And there are 2 most common, secondary structures, the alpha helix and beta pleated sheet. So for the alpha helix, the protein backbone is coiled and stabilized by hydrogen bonds between the amide and carbonyl groups. And then the other, beta pleated sheets are just peptide chains that are fully extended, not coiled, and then there's hydrogen bonds between the adjacent chains. For tertiary structure, that's just the protein's 3D shape. It depends on hydrophilic and hydrophobic interactions of the polar and nonpolar side chains. And it's good to note that denaturation of the protein occurs at this level when there are changes in temperature or pH, and this is due to the tertiary structure being stabilized by weak intramolecular interactions, which can be easily disrupted. And denaturation means that the primary structure of the protein is unchanged, but the tertiary structure will unfold and become randomly looped. So it can cause changes in solubility and catalytic activity in the case of enzymes. And it is usually irreversible, but there are some instances where you can reverse it. So for quaternary structure, that is just multiple protein molecules combined into one structure. So enzymes are substances, usually large proteins, that can act as catalysts for biological reactions. It doesn't affect the equilibrium constant of the reaction, but it actually lowers the activation energy instead to make the reaction happen much more rapidly, sometimes up to 1,000,000 times faster. So unlike many catalysts used in chemistry labs, enzymes are extremely specific and usually only catalyze a single reaction of a single compound, which is called the substrate. And enzymes are able to accelerate reactions so quickly due to factors such as simple geometry, because the enzyme will adjust its shape to hold the substrate or other catalytic sites. And it will form the exact geometry needed for the reaction. And also another factor is that enzymes will stabilize and lower the energy of the transition state of the reaction, which would cause the reaction to accelerate. So we've talked about the protein parts of enzymes, but most enzymes also contain a small non-protein part called a cofactor. It can be an inorganic ion or a small organic molecule called a coenzyme. Coenzymes are reactants, not catalysts, that undergo chemical changes during the reaction, and they are derived from vitamins, and they also require an additional step to return to their initial state. And some examples are like coenzyme A, which is derived from vitamin B3, and FAD, which is derived from B2. And this figure on the right just shows kind of the role that coenzymes play in, formation of the enzyme substrate complex. One application of this would be alcohol metabolism. And we start with alcohol dehydrogenase, or ADH, which is an enzyme that begins the process of breaking down ethanol in blood. The name dehydrogenase suggests that it is oxidizing the ethanol by removing hydrogens from it. In fact, as we see in the figure in the upper right corner, this enzyme actually has a coenzyme NAD+, which is very important in this reaction. And so NAD+ plays roles in not only redox reactions in cells, but also key cellular functions such as metabolic pathways, DNA repair, and chromatin remodeling. And so we see that NAD+ is converted to NADH, which ends up leaving the active site along with the product acetaldehyde, which is very toxic to the human body. So it is quickly converted to acetate, which is less toxic, with aldehyde dehydrogenase. And we see that in the lower right corner. And this aldehyde dehydrogenase, or ALDH2, actually uses NAD+ as a coenzyme as well. And then so after a series of enzymatic reactions, we get CO2 and H2O for alcohol metabolism. Some people will have ALDH2 deficiency, characterized by a red complexion or glow after drinking alcohol. And this is actually a genetic mutation in ALDH2 that causes extremely low enzymatic activity during alcohol metabolism, as low as 4% compared to normal enzymes. So because ALDH2 catalyzes the reaction of acetaldehyde to acetate, this can lead to an accumulation of acetaldehyde, which can be very toxic to cells. It can actually lead to mutations in DNA if the levels are above 50 micromolar, and it is known to be localized, so it can cause a lot of effects in the mouth and throat, and also increase risk of head and neck cancers. And then the figure on the right just shows the difference between wild type or normal ALDH2 and mutant ALDH2. And we see that there is very little or no acetate production, which will just lead to a lot of ALDH2 accumulating. So yeah, that's it. Thank you for watching.